Hello, my name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce Dr. Fabio Luciani. Dr. Luciani is an Associate Professor and Senior Researcher in Systems Immunology at the School of Medical Sciences and at the Kirby Institute, University New South Wales, Sydney. He has a PhD degree in Theoretical Immunology and Biophysics from the Humboldt University of Berlin, Germany. He's a theoretical physicist where he received his master's from Bologna University, Italy, with training and research experience in immunology, genomics, statistics, computational biology, and bioinformatics. His current research focuses on adaptive immune responses and cellular therapies against viral infections and cancer utilizing single-cell technologies. He has developed and applied single-cell genomics and systems immunology approaches to understand T-cell dynamics. With that, I turn this webinar over to Dr. Luciani to present his talk titled Applications in CAR T-cell Therapy, Dissecting Cellular Composition Using Single-Cell Multiomics. Thank you.
Thank you, Jeremy. Welcome, everyone. Today, I'm going to share with you some recent work in our laboratory on the application of single-cell multiomics to study CAR T-cell therapy. The goal of this presentation is to dissect the cellular composition, as the title says, of this therapy and using the technology to unravel the heterogeneity and molecular signatures of those cells. I'm going to just move now to next slide. In this presentation, I will break up the points into 5 major items. The first thing I will present is an introduction to single-cell multiomics in general, and the particular one that they have used in this study from BD. And I will also introduce CAR T-cell therapy for those of you that might not have a good knowledge of this. Next, I will move into some experimental design and the analysis we have performed, and then I will move into bioinformatics analysis, and will give you some basics and details of what we have done, and then I will go through the applications, and then finally I will discuss some conclusion and recommendation from what we have learned from this experience. As mentioned, CAR T cells are basically CAR stands for Chimeric Antigen Receptor T cells, and those cells have been come to the fore recently in therapies for blood disorders such as lymphoma, and that's one of the focus of CAR T-cell therapy, although there are other CARs around now to trial towards solid tumor. In this slide, what I'm showing is a very simple as it can be, of CD19 specific CAR T cells. So effectively T cells that have engineered to target B cells in the context of lymphoma. On the left-hand side, you can see this little draw of a normal T cell, this blue cell, which has a receptor, T cell receptor, that targets a small antigen on the surface of antigen-presenting cells, in this case tumor cells, this orange cell, and then this is a specific recognition of T cells towards the peptides MHC complex. CAR T cell follows a very similar principle. However, CAR T do not recognize peptides in the context of MHC. In this case, CAR T are engineered to target directly a domain of interest, in this case, CD19. So it's an application of engineering cells to target a specific molecule on the surface of cells, and in this case, B cells. These therapies are now in several clinical trials, as well as have been approved by FDA, for instance, to treat blood disorders such as lymphomas. And the way this therapy works is basically cells are collected from a donor, as you can see in this slide, point number 1. Those cells are then engineered to via transduction, for instance, or transcription of specific receptors that can target your B cell, for instance, and then they are produced as in large amounts and those are eventually adoptively transferred to the patient. And then patients have to be monitored to check whether those CAR T are effectively proliferating, expanding, and effectively killing the tumor. And that's the space, number 4, where we have been focusing on with this technology, which I'm about to explain. So in other words, because CAR T cells are adoptively transferred and undergo dynamics, so they proliferate, they can die eventually, and they can defeat the tumor, they need to be monitored, and they, because they evolve, we need to understand how they function on a molecular phenotyping are actually changing during therapy within the patient. And as you can see, a little cartoon on the right-hand side, just a little animation to see how this specificity occur, where basically T cells recognize a molecule in the context of a tumor, and then they release cytokines to kill the tumor. So as I mentioned in the previous slide, in order to identify the molecular or genetic signature of those cells, we adopted an approach that utilized single-cell technology, and specifically a multiomics approach. Multiomics here stands for the possibility to take from each individual cells different measurements. And in this particular case, we are measuring the transcriptomics, so the mRNA within the cell, as well as a protein expression on the surface of those cells. The technology we've used is the AbOligo from BD, also known as AbSeq, where effectively we conjugated antibodies who have attached to them some oligos, and those antibodies are stained, so they get in reaction with cells, and then those cells will, if they have the specific receptor, will have those antibodies attached to the surface. Those cells are then put into BD Rhapsody machine, which is a machine that permits a capture of single cells on plates, which I will show you to the next slide. And then effectively what you're able to do with this technology is to measure simultaneously for each single cell RNA from the messenger RNA inside the cell, so the transcriptomic profile, as well as the surface staining, the surface profile of those cells. Why this can be done in such a large number of cells is because those antibody that we stain, as well as the mRNA, have barcodes inside those molecules, which allow us to demultiplex bioinformatically whether those measures that we do come from a certain cell or another cell. And this is the technology that we refer to as multiomics. In the next slide, this is a little cartoon explaining how exactly the BD AbSeq oligo works. So you can see you have an antibody, the classical antibody that you will use, for instance, in classical flow cytometry. And then this antibody has attached a number of sequences, a PCR handle, which is a common sequence to allow for the binding of the antibody to this oligo, and then a unique molecular identifier, UMI, which allows basically to have enough diversity to be able to demultiplex many molecules within each single cell. And then you have a barcode that will identify the specific cell. And you have a poly-A tail. That's effectively to basically allowing to interpret this antibody as a sort of mRNA molecule. And in that way, this will be treated as if it was an mRNA molecule coming from inside the cell. After we stain it with antibodies on the surface of the cells, as I mentioned before, those cells are captured into a BD Rhapsody system. And this machine has a chip inside, a cartridge with microwells, and cells are gently deposited on those microwells, and then via certain washing and few other steps in protocols that are available online, you can isolate between 1,000 and up to 10,000 cells into those wells, and from those cells there is a protocol that allows you to convert into cDNA and then sequencing those, amplifying the sequences those cDNA, and then you get results for each single cell. The advantage of this technology is that it does not use microfluidics, so they might not really clog cells altogether. It's very gentle on the cells. And then you can visualize whether the cells are into the wells or not. And you have a magnetic barcodes that can be retained for later experiments that you want to perform. So that was a summary of the single-cell multiomics and CAR T-cell therapy, just to give a background. The next thing I'm going to show you now is what we do with these experiments in the context, with this technology in the context of CAR T-cell. So I'm going to show you the experimental design and the overview plan that we have performed. The BD AbSeq markers we have used in this particular experiment was a collection of 40 markers. And as you can see in this table, we have chosen markers keeping in mind that we were looking at the blood of humans, and therefore we have looked at lymphocytes and monocytes circulating into the blood. Therefore, we have selected markers that will allow us to characterize lymphocytes as well as their differentiation status, whether they are active, whether they are exhausted T cells, for instance, as well as there are B cells. And we can also looking at co-inhibition and inhibitory markers such as PD-1 or TIM3. And then we also moved into 7 markers that will allow us to characterize in more details the monocytes. And for instance, we have CD14, and then we have considered CD11B and CD11C. And we can also characterize dendritic cells, as you can see, by markers of CD141. And as you can see, several other markers. So the idea was to use our best knowledge we have to use these AbSeq markers to characterize every single type of cell that we would encounter and measure into the blood. With these markers, then, we have taken blood samples from CAR T patients, and we have taken peripheral blood, PBMC, as well as we have sorted from this blood also CAR T cell specifically. Therefore, we perform this technology on any blood cells as well as only on CAR T cell. And of course, I will tell later on a few applications in these 2 experiments. After the cells will be sorted and they get captured into the Rhapsody system, as you can see in this slide, and then sequences are generated by next generation sequencing, and then the bioinformatics is performed to identify protein level on the surface of the cells, as well as gene expression and a few other things which I'm about to explain. So that was the experimental design and the overview of how we have used this technology in which sample. The next thing I want to explain is the bioinformatic analysis of this high throughput data. So I forgot to mention, but these data are large, of course, they're big data. So for each cell, we have the whole transcriptome, as well as these 40 surface markers. And we have performed this on multiple sample time points. Therefore, there's quite a lot of bioinformatics that needs to be done in terms of checking the quality control of the data, as well as then the application of those data to understand the biology. The bioinformatics, I will briefly go through the major steps, which are available for reading later on. The BD AbSeq and gene expression counts can be obtained actually by an analysis pipeline, or whole transcriptome analysis pipeline, available via the cloud on the 7 bridges, as well as you can install locally in your system. Therefore, if you are expert in bioinformatics, you can do your own analysis, or there is a cloud system that allows you to obtain those counts. The next thing we perform once you obtain the counts is to do a QC step to look at the analysis of whether the data that we have generated makes sense. In this case, what I'm showing you is a typical first step analysis of what we obtain. This is a typical output of gene expression where you see on the x-axis on the first plot, you can see is the total count, so how many basically reads we generate, and then how many genes you can measure with that count that you have. And you can see, of course, that the more you sequence, the more genes you detect, and then eventually this curve started to saturate a bit. You have to check, however, whether within those cells there is a lot of mitochondria genes, because those above a certain threshold might indicate that your experiment didn't perform very well. Sometimes it happens in high throughput single cell analysis, so you might need to exclude some high amount of mitochondria in some cells. And as you can see here, we adopted two thresholds, so you can see that there is a high mitochondrial ratio for very low counts of cells, which means some cells didn't really have enough material sequenced, and therefore there is some overexpression of mitochondria and not much else, and therefore those cells will be removed. You can see then the same thing by plotting on the x-axis the mitochondrial ratio, and on the y-axis the unique genes. And so you can see again that when you have a lot of mitochondria, so the ratio approaches to 1, you can see that there's no many other genes you're actually measuring. So that allows you to make a judgment on the quality of your data. And it's very, depending on the question you're asking, on the technology you're using, this threshold can be moved around. More importantly, you probably have heard in single-cell transcriptomics field, the genes might have this zero expression inflation. So genes that you would expect to be non-zero, but you measure zero value for it. And those are commonly known as dropout. And there are technology out there and bioinformatics tools to deal with that and to infer dropout rates. The next step we do after the QC is the normalization. So cells, you can assume the cells have an equal number of mRNA molecules between them. So the simplest thing you could do, if that's the assumption, is that you divide everything by the total count. That will allow you to basically compare one cell with another. You can, however, looking at other variables that might affect the quality of your data, such as the library size, and you might have experimental conditions that might have affected the quality of your data. So you could think of more complicated models. For instance, you can consider a multivariable model where you assume the cells are not actually as similar as you would think, and therefore you need to account for potential batch effects that might occur in your experiments. This is very common in blood samples from cryopreserved tissue or blood, like in this case, because, of course, obviously cells are stored at different times from different patients, from different time points, and the analysis performed on different days. Therefore, it would be crucial to consider all those variables to make an appropriate analysis. And there are things out there, such as transformations, that could help you to visualize the data and to look at whether your genes are following an appropriate distribution, statistically speaking, or they have a skewed distribution, such as a lot of dropout, as I referred before. And I'm more than happy to answer questions about this. The next thing is to then look at the data. Once you believe your data are clean sufficiently for downstream analysis, what you can do is to start to look at the output. And in this case, is the AbSeq output, where you can see what we have done here is to perform the experiment with the AbSeq oligos. On the left-hand side, in this case, CD8 and CD3, you can see basically cells that are CD8 positive, CD3 positive. So those are the CD8 T cells. Then you can see the CD3 positive, CD8 negative on the lower quadrant, and there's a CD4, and then you can see some double negative as well. So this CD3 negative cells, those could be, for instance, monocytes. And interestingly, what we have done a parallel experiment where we have performed from the same sample, we took an aliquot of cells and we looked at the classical flow cytometry. So you can see that the distribution in flow cytometry is very similar to the one that we obtained in AbSeq. So again, suggesting that this technology are going into the right direction, and despite our incredibly high throughput and with the barcoding system, which can be prone to errors due to the sequencing technology we are using to take this output, the results are actually pretty consistent with flow cytometry. You can see the same thing on the bottom quadrant where you look at CD45 array versus CCR7, which allows you to have the canonical distribution of T cell subsets, memory, effector, naive. And those cells, you can see again the distribution between the AbSeq measurements on the left-hand side and the classical flow cytometry are again quite consistent. And you can see the numbers. I know they're very small, but probably you'll be able to see them. They are quite consistent to each other. For instance, you can see that there is about 4.7% of CCR7 positive CD45 array positive cells, and that you have a 3.4 cells. So very close number to each other, suggested that this technology are indeed going into the right direction. After we have confirmed so that these data we have generated have a good quality and they can be comparable to what we know already, then we could start to explore this in depth of the high throughput that we have generated, so 40 markers on the surface of the cells and the entire transcriptome. And this will be done, we'll show you a few applications of based on the first question that you can actually ask with this technology is that how many cell types we identify and what is the gene signature associated to each of those types. As a first example, let me introduce you to this classical way of representing high dimensional data. You probably have come across these dimensionality reduction plots if you are interested in single cell technology. This is, in this case, is a UMAP plot, very similar to a t-SNE plot or a principal component analysis plot. So effectively, we are reducing in 2 dimensions the complexity of each cell. So each dot in this plot is a cell, and as you can see, in these 2 dimensions, we are able to characterize different types of cells. So, and what we've done here, we're just taking the AbSeq oligo data. So we're just taking the expression level of those 40 proteins on the surface of the cells. We did not use the gene expression, so it's a single omic here, and we asked the question, can these 40 markers at the single cell level identify the cell types? And the answer was yes. So in this particular case, we take PBMC from sample collected 49 days after infusion of CAR T cell in this patient. And so you ask the question, what happened 49 days after we infuse CAR T? And I just want to remind you that these people that undergo treatment are immunodepleted to start with, so their immune system is almost close to a very small number of cells. So what we are expecting is that the immune system will eventually go back with the CAR T, but also any other cells of the we started to reappear into the blood. And what you can see here is that I color-coded all the cell types. So you can see there is CD4 cells at the bottom, the red ones. There's this massive population of CD8 positive T cells. There are some other minor populations. For instance, you have the monocytes on the top right quadrant. And then there are some other ray cells, such as dendritic cells of NK cells. And in yellow, there are B cells. You probably can't see them very well, but the B cells are almost absent here, and simply because CAR T cell have been effectively in targeting their antigen, which was CD19. And therefore, we were not expecting a lot of B cells into this sample, and that's exactly what we found. With this in hand, then we asked the question is that can we learn more about those subsets by using the other markers that by looking at one by one all the 40 markers, what those markers tell us in terms of further characterizing eventual subsets that might be within those major clusters. And this is what we've done here. We have taken that same experiment as before, but this time instead of coloring by cell type, we are coloring with a scale from low to high, from red to dark, from purple, of individual markers. So in this case, for instance, top left, you can see CD3. So CD3, you will see is yellow for the monocytes, and started to be blue and sometimes purple for lymphocytes, for the CD4 and the CD8 T cells. And that makes sense. So you can see how the CD3 marker identified the right population. You can move then to the CD4, and you can see that the CD4 is blue on the CD4 cells, of course, and so on and so forth. And you can go then into more details. For instance, you can looking at HLA expression, CD14 for the monocytes, as well as CD16. And that gives you an idea of the actual heterogeneity in the expression of each single marker in each single cell. The next slide shows you now some extra information you can obtain from AbSeq oligo staining on those cells. This time, instead of having one sample, we have analyzed 3 samples. So we asked the question, how is the immune system evolving over time during this therapy? So on the left-hand side, you see again a UMAP plot from a pre-infusion condition. So this is the blood of the patient before receives CAR T cell, but after immunodepletion, which means its immune system was severely depleted. And in this case, as you can see, there is only 3 major classes of cells here, which are effectively some CD4 cells in red, and then you can see some CD8, and then we had this light blue here, this major population here. Those were mostly monocytes. And interestingly, 7 days after you infuse CAR T cell, you can see how the immune system is tremendously changed. So you can see now there's still CD4 cells in red. However, you can see this massive expansion of CD8 T cells in blue. And then again, the monocytes are there. However, we found much less monocytes here. And notably, we found this green population. This green population is a CD3 positive cells who lacks the expression of CD3 and CD4. And we have been wondering what this population was. And you look at day 49 on the right-hand side at the same type of plot, the UMAP, and you can see that this type of population keeps evolving. And you can see how monocytes keep shrinking and T cells, CD8 T cells remains there, and you see CD4 in red expanding further. And the message of these slides is basically what we are learning is that within 49 days from the infusion of CAR T, you can see a significant increase of CD8 T cells overall, as well as a decrease in monocytes. And this is just one application of what you can do with the AbSeq oligos by just looking at 3 sample time points. Next question was, can we look at subtypes of, for instance, certain type of cells, such as CD8 T cells? Can we look at subsets of those cells with our markers, with the AbSeq marker? And what we found here, you can see how we are going from pre-infusion again to day 7 and day 49 of the canonical markers of central memory, CHAM, naive, effector memory, EM, and TEMRA. So you can see in an immunodepleted condition, pre-infusion, you see there's not many CD8 T cells going around, but then just 7 days after that, you can see an expansion of TEMRA and effector memory cells, and then at 49 days after infusion, you see the immune reconstitution effective with all the population coming back into game. And in this case, what we observed was an expansion of effector memory array positive cells, and as well as but interestingly, we saw a decrease in effector memory cells. So these are just examples that what you can do in terms of going into details of subset of cells by just a single experiment. The next question was, what happens if we now consider not only the omics, the expression level of proteins that we measure with AbSeq on the surface of each cell, but what if we now introduce the other set of information that we have, which is the whole transcriptome analysis. So for each of those cells, we have also the gene expression amount of each gene expressed in each single cell. And how can this information then now be integrated to the AbSeq information that I just showed you now? And what does it tell us? What can we get by integrating a second omic on these experiments? And what I'm about to show you is some application to discovery, for instance, a new cell population. So in this experiment here, you might recall you have seen this picture just a few slides before. This was again a UMAP plot out of a patient that, and this was 7 days post-infusion. You remember I mentioned to you there was this green population of CD3 positive cells which lacked the expression of CD4 and CD8, and they were from an AbSeq point of view. So this was the picture generated just with the oligo. With those 40 markers that we measure by AbSeq, you could see this green population. And the question was, what are those cells? Can we use gene expression to identify more details about this population? So that's what we did. So what we have done, we have taken a UMAP plot from the AbSeq. And the next thing we have done, you can see we have taken just 2 genes from Rhapsody. So not the whole transcriptome, just 2 genes. And we asked the question, what if those cells are gamma delta T cells? because we knew they were CD3 positive and they were lack CD4 and CD8, so we had already an indication that those cells could become, could be some sort of gamma delta T cells. Although there were very, very large amounts of those cells in the blood, and we were not expecting that, so it was a bit of a surprise. What we have done, therefore, is to look at the constant region of the delta chain and the gamma chain that was available by Rhapsody analysis. And therefore, if we would see the simultaneous expression of a delta and a gamma, that was a genetic signature that, you know, the expression of those receptors in those cells would strongly indicate that those were gamma delta T cells. And as you can see now, what we have done, we're colored in red everything else, and we are, so no expression. And then what we said is that, can I see a delta chain? And as you can see, as I circle here, you could see cells expressing the delta chain constant region of the delta gene as well as of the gamma gene, again suggested that that population was indeed, within that cluster was definitely some gamma delta T cell. So there was basically a discovery we performed, and this is about 16% of the total cells that we are looking at these plots. And so there was a discovery that after CAR T cell infusion, just 7 days post-infusion, you can obtain up to 16% of gamma delta T cells circulating into the blood. Now, of course, the next question would be, what are those cells doing? And of course, we are undergoing further experimentation to characterize those cells. A second application of the power of integrating transcriptomics analysis with the AbSeq marker is actually to obtain the gene signature of those subsets. As you can see here, there is a heat map of the same data that I showed you in the slide before, but this time in each column we put a cell. So in this heat map, every column is a cell, and every row is the expression pattern of certain genes that identify those cell types. So you can see there is a band of colors on the top of this map. Those are the 5 clusters we identified by performing a supervised clustering here. And what you can see is that we can identify CD8 T cells on the top left with genes that are known to characterize CD8 T cells. And then you can see the CD4 in cluster 2, and then the cells that I've just mentioned in the slide before, the gamma delta T cells, and with some interesting gene patterns of definitely confirming that those were gamma delta T cells. Then you can see the monocytes as well as the NK cells. And there are some known and unknown gene signature here that we have discovered, which we are now undergoing further experimentation to understand even further the heterogeneity within those 5 populations. And that's a very straightforward application of this technology in this type of settings. The next thing I want to show to you is actually a little bit more complex analysis where you can actually take your CAR T cells and the total T cells that you have in the blood and ask the question, how those CAR T cells compare to the normal T cells that are circulating in the same blood at the same time point? So as you might recall at the beginning in the experimental design, I have considered 2 setups where we would use single-cell multiomics on the total peripheral blood mononuclear cells, but we have also sorted specifically CAR T cells from the same sample. Therefore, we do have a signature of CAR T cell, and we do have the total blood. So we can ask the question, how does those CAR T cell compare to the CD8 T cell, for instance? So you can see here again a UMAP plot of from the total blood, which is the top left. You can see this massive CD8 population, as I mentioned before, the blue one. And at the bottom, you have a UMAP of actually only the CAR T cells, and in particular, only the CD8 CAR T cell, because there is also CD4 CAR T cells. But in this case, I'm showing you only the CD8 T cells. And the question is, what are those 2 populations have in common? So to do that, what we have performed is a bit more of a complicated bioinformatics analysis where effectively what we have done, we have the word that we use now is harmonizing. So we're basically integrated those 2 data sets together in a single UMAP plot. And by taking this time the gene expression and not the AbSeq marker data. So the gene expression shows you this now these cells integrate, and as you can see, there is clearly a batch effect because the CD8 T cells and the CAR T that are CD8, they definitely separate. And it's probably very likely due to the batch effect of those being two distinct experiments. So what we have done is to perform an integration of those samples together by performing a mutual nearest neighbor approach, which has been previously published and available in several pipelines. In this case, we have used the pipeline Seurat, freely available for those of you who want to enjoy a bit of bioinformatics. And you can see now those cells can be integrated within the same scale. The gene expression level now are compatible to each other. And therefore, you can see now that what we have is 3 clusters of cells. Some of those clusters contains both CD8 T cells and CAR T cells. For instance, you can see how the green cluster at the bottom left is actually a sum of the previous slide of the pink and the blue, which were basically CD8 and CAR T cell. And then you can see up on the big cluster on the right-hand side, the red one, you can see is also a combination of both CAR T cells and CD8 T cells. So you can then ask the question, what is the gene signature that characterized each of those 3 clusters? And that's the next slide. So what we have done, now that we have integrated CAR T cell and CD8 T cells, we said, okay, what is the question? What is the gene signature differing between them? So you can see again those 3 clusters. I just named them 0, 1, and 2. Sorry. And you can see that the cluster 0 is the largest cluster, which is characterized by granzyme H and KLRD1, for instance, and MALAT1. And then you go to the green cluster, cluster 1, where you can see that there's more expression of tuba and some other genes like PCNA and so on and so forth. So you basically discover a specific signature that characterizes subsets of cells circulating in the blood and tells you also what do they have, which gene characterizes each different clusters, and therefore how the CAR T cell are differing from the total CD8 T cells circulating in that blood. So this is a very powerful technology to identify to an unprecedented level subsets of T cells that are evolving during therapy, and also to ask the question of how those engineered CAR T cells are differing from the normal T cells of that patient, and what could be the interactions between them. So this is a very significantly advanced technology. We are still analyzing this data. As you can probably see by looking at these slides, there is a lot of other questions you can ask and specifically other consideration you could do. For the purpose of this webinar, I would just stop it here just to give you ideas of the power of those technologies. The last thing I want to recommend is to sort of summarize what we have learned from these applications, and also tell you what we're doing right now in the lab. So in summary, what we have learned here is that single-cell multiomics is an effective way to comprehensively understand both the cellular composition as well as the molecular composition of samples that are available, for instance, from clinical trials that are cryopreserved. So those samples can be used and characterized to an unprecedented level, and they can provide you significant in-depth analysis of molecular and genetic signature of those cells that are characterizing those samples. You can identify blood cell types, and along with those, the gene and protein composition, and how those actually evolves during therapy, for instance. This is a powerful approach, of course, to also identify differences of, in this case, of engineered CAR T cells compared to actually the normal T cells that are into the blood, and ask questions, for instance, of how those cells interact. Do they interact at all? Do they share any signature? What can we do to improve CAR T cells to make it not look very different from the rest? And can they become memory cells? Are they looking the same? And then you can identify also the evolution of those CAR T cells during therapy. For instance, you can consider scenarios where CAR T cells are lost, so they undergo exhaustion, for instance, or CAR T cells that survive and they might provide long-term remission to patients. And on a large scale, of course, these methods can be performed to identify subtypes, gene and protein composition, and on a very, very large number of samples, and therefore you can look at other translation application of this technology. Future directions, at least in our lab, is, as you probably have appreciated from this presentation, most of this data requires a significant know-how on bioinformatics and integration of data sets together, so what we call harmonization. This is a focus in the lab in the next few months. The other point is we have a lot of interest in specifically T cells, and in CAR T cells there is another level of omics that you can introduce, which is actually the clonality of those cells by identifying the T cell receptor of those cells. So we have some measurements going on now on the same samples where we add the third omics to the gene expression and to the surface protein expression, which is the T cell receptor, and we are integrating the clonality with the gene expression as well as the protein expression. So in summary, what we are aiming is that to bring single-cell multiomics to discover more, so basic knowledge, but we are also aiming at translational application of this technology. As the cost will go down and this technology will improve significantly in the next few months. So finally, I would love to thank the people that have really done this work, and that's our two people in my lab. One is Curtis, which is a PhD student, about to finish, and Dr. Ray Louis, who is a postdoc with a significant bioinformatics expertise. I would like to thank also the Westmead Hospital, in particular Dr. Ken Micklethwaite, who is the PI of the clinical trials of the CAR-19 T cells we have used at the Westmead Institute. And I would love to thank also the Garvan Institute colleagues, Dr. Manu Singh and Katrin Jackson, for analysis of and support on the bioinformatics analysis, as well as, of course, BD that have kindly supported this project. So I'll leave it there. Thank you so much for listening, and I'm here opening to take questions. Thank you.
Thank you, Dr. Luciani, for that informative presentation. So our first question is concerning what is the effect of cryopreservation on the quality of scRNA-seq and AbSeq data?
We have performed some experiments to compare some fresh sample versus cryopreserved, and it is very variable on the sample that we analyze. However, generally speaking, on average, our results shows that if we isolate live cells after thawing the cells from the cryopreserved samples, results are very consistent with fresh samples. The variability, of course, is higher. Therefore, my advice would be to say that it is possible, absolutely, to generate high-quality cells and data. However, we need to be careful about the way we handle the samples and the quality with which we select live and dead cells.
Thank you. Are CAR T cells significantly different than normal T cells in terms of their gene expression and surface expression?
That's a very good question. It is the question that we always ask since the beginning. It is interesting to show, as you can see from the presentation, that CAR T cell shares some common signature with the other T cells that are circulating into the blood during CAR T cell therapy. However, we do see a rapid onset of very active T cells with a very cytotoxic profile, definitely above the profile that normal T cells have into the blood at the same point in time within the same sample. So the power of this technology is that you can directly compare CAR T versus normal cells. So the results so far, based on what we have seen and needs to be validated, of course, on other patients and samples, is that there is a shared signature to the other T cells, which are also showing active profile, but it is absolutely clear that there is a subset of CAR T cells that share a unique signature. What that means, of course, now requires further investigation, but it's definitely a good question.
How can one control for batch effects and cell doublets using the AbSeq and Rhapsody platforms?
And that's a very, very interesting question, and it's one of the hot topics in bioinformatics for researchers and that are working on single-cell multiomics. Batch effect is an issue, and it is known to be an issue for single-cell multiomics and single-cell transcriptomics in general. When you perform single-cell multiomics, therefore, you will encounter the same issue that normal single-cell transcriptomics, for instance, experiments will show. How do you handle this? Well, there are two different ways to do it. So, bioinformatically speaking, you could definitely interact more with the experimental people and make sure that the conditions of the experiments they do contain controls and specific designs that you can do that will help the downstream bioinformatics. And the second approach would be to perform analysis of the data in terms of normalization, and statistical tools exist to also correct for very clear and evident batch effect. For instance, one example would be if you perform analysis across multiple samples that are obtained from different subjects, it is very likely that because those samples have been analyzed at different point in time, probably by different users, then there would be that effect. And usually what results is that sometimes experiments generate a higher library size than others. This is, however, can be easily corrected because if you have two experiments with a high and low library size, and with library size I mean number of genes identified, it could be different coverage, for instance, then you can kind of normalize these two samples together with standard bioinformatic tools that are available in many of the open source softwares. In this particular one, we used Sura. There is a very, very helpful vignette. And so it's very easy to approach in this way. But however, I would generally conclude saying that it is very wise that bioinformaticians will stay in close contact with experimentalists and try to design the experiment in a way that the downstream bioinformatic analysis can be very powerful in controlling from batch effect. Hopefully this will answer the question. But I'm happy to answer further if people want to email further.
So that actually leads into the next question, which is, can you provide comments further about some of the specific bioinformatic methods that were applied to integrate protein and gene expression data and to identify cellular composition?
Yes, that's I hope I conveyed a bit the message here. This is as I mentioned at the end of my slides, this is a hot topic. The way we're doing at the moment, as I mentioned, as I demonstrated into the slides, is that a simple approach would be to the simplest thing we could do, which is also recommended by, for instance, some available packages, such as Sura, is to first analyze the protein composition and then analyze into transcriptomics. In the way we did it, we basically integrated the data by looking at the gene expression patterns that characterize specific subsets of cells that were identified with AbSeq. So in other words, you can use one technology to identify cell types or major family of cell types, in this case, CD4, CD8 T cells as well as monocytes, and then you go in with the gene expression profile to identify further heterogeneity. Now, the future probably will be in a very short time. There would be some more advanced statistical tools where you can simultaneously analyze gene expression and protein within the same object. This, however, at the moment, it's still under investigation and results are not really final. So I would recommend that the best way to integrate multiomics is to basically make those omics interact to each other, but making sure that you have thoroughly analyzed individual omics first. So analyzing transcriptomic alone, AbSeq alone, and then you start to integrate them together. It's cellular composition at the moment. It can be done by single-cell transcriptomics alone or AbSeq alone. So I guess the advantage of single-cell multiomics, even by doing what I've just said, will allow you to go even deeper into cellular composition and identify novel cell types, which probably without multiomics cannot be identified.
Thank you. What is the lowest frequency of cells that may be detected by AbSeq, and can it be used for applications with rare events?
That's another very interesting question. So the rare cell types is something challenging to identify. You can definitely use this technology to identify types. However, if those cells are very rare, then there is alternative solutions. Like one solution is the one we performed here, where you pre-sort the cell of interest. In this case, we're CAR-T, right? If we were to just look for CAR-T in the PBMC sample, the chance to have a single CAR-T cell out of, say, 5,000 cells would have been less than 1%. Therefore, those cells have to be enriched a priori. There is other ways, however, you could do. You could also use the multiplex technology available with AbSeq, where you can combine multiple rare cells that you can sort upfront in flow cytometry, and then you barcode them in the same multiplex into the same run, and then you can look at that way. At the moment, those are probably my recommended ones using this technology. In the early future, I'm sure BD will release a lot of more advanced technology, and there will be possibility to perform even more precise identification of rare cell types. I guess that's really what we're after at the moment with this technology.
Thank you. A kind of a follow-up question to that is, what is the magnitude and nature of the difference in gene expression between the CAR T cells and the native T cells? Was this something that you expected to find, or did it come as something of a surprise?
It's exactly like I kind of already mentioned a little bit on the previous question. No, I was surprised, right? And I was surprised in both ways. We did expect some sort of difference between CAR-T and CD8, but what we found with this technology is that, first of all, even the total T cells, the CD8 T cells in the blood, in this condition under CAR-T cell infusion, looked substantially more active population than you would have expected. So somehow the presence of CAR-T have disrupted the gene signature profile of the entire population of T cells. That was our first surprise. The second one was that we said, okay, so maybe then CAR-T and CD8 are all the same. And as I mentioned in the question before, I said no. As you can see in the heat map that I showed before, there is even a subpopulation of CAR-T cells that are even different from the ones that are present in the blood. So I guess that to summarize this question, hopefully I will make it clear enough. The fact that we have used single-cell multiomics across both CAR-T cells and normal T cells was probably a very good thing to do, because if we would have done only CAR-T, we would have never discovered the similarities and differences with the other ongoing populations. And the same thing, by the way, holds for other cell types, right? So it's not only T cells, but also B cells in case they come back, in case of relapse, patients that relapse to CAR-T therapy, or also looking at monocytes response and the effect of those in cytokine release storm. So it's a very, very interesting way to explore uncharted territories on how T cells and other cells of the immune system evolves during therapy.
Thank you. One of the questions is that if someone looking at ways that you can use this technology is if they do not have a panel of antibodies, is there a way to modulate this technology and use it without antibodies?
So I'm not sure if I understand the question about the antibodies for AbSeq technology, you mean?
I believe so, yes.
Yes. So you could, if it's about flexibility, yes, you can be quite flexible. You can remove some of the antibodies. For instance, sometimes, if you don't see a cell population or you have too much of a cell population that express a certain marker, you could remove that marker as well, because that would probably be sequenced as the most abundant antibody. You can add antibodies. You can perform just with a minimum number of antibodies if you can. We have done that as well. And of course, probably, I don't know whether that is what the person that asked this question was referring to. You can also go back to individual omics, of course. The point of this, however, was that you could combine these two technologies together, and you can be flexible on the type of and numbers of antibodies that you use to stain the cells before Rhapsody. So hopefully this will answer the question.
Thank you. So you did mention being able to remove populations and look at different populations. Do you have any recommendations about how to deal with sample heterogeneity issues?
Yes, heterogeneity is it's an interesting topic. As I just said in the previous question, there is no way to say a priori for most of the cases whether there is a very, very abundant population and a very, very rare population, and whether or not your markers that you're using in AbSeq are good enough to discern these populations. However, in the case of the blood, we do know quite well the general composition of cells circulating into the blood. So the way we designed this antibody panel for AbSeq was to ensure that we would cope to in scenarios where the rare population were small enough but yet still measurable. So one example would have been B cells, where we knew that CAR-19 cells were killing B cells. And as I show in one of the slides, probably wasn't very clear, but there were some yellow B cells around that. Those were probably B cells that were not being killed by CAR-19, and we could still see them, right? So I guess I can answer with what we have done, but with the setup of the panel that I showed, we can identify rare population just with the AbSeq antibodies and in the blood. Whether or not this is going to now be true with tissue, there are some bioarchive papers out there that have used AbSeq technology, for instance, in gut tissues, and by reading those papers, the results are very consistent with what we found into the blood, namely that there are rare population, for instance, ILCs, subtypes of ILCs that can be identified with actually a similar number of antibody markers. So my message is that I think we have a good setup to go into uncharted territory of looking at heterogeneity. But if you then are interested in specifically certain populations, then I think you should go into the approach that we have done, where you pre-sort the cell of interest, like in this case, CAR T cell, and then you will thoroughly analyze that particular subset.
So CAR T cells have been looked at for the use of treating other different types of disorders. So can this process be used as research for other types of cancer in the body, such as colon cancer, liver, or lung cancer?
Yeah, so the CAR T, outside the technology, yes, the CAR T are now exploding. So there are about more than 300 clinical trials and counting around the world. They are targeting about 60 to 62 different antigens. There's several papers out there, including reviews, a new database is set up, which you can look for in literature. Most of the results that we know are about blood disorder, because that's where the CAR T have been already commercialized with several products into the market. Several trials, however, are undergoing for the treatment of solid cancer. This technology actually have been already implemented in some of those trials, including we are doing some collaboration with other colleagues in the U.S. On solid tumors. So I can envisage that with the rapid growth of those clinical trials and the importance of investigating the entire arms of the immune system and the tumor heterogeneity, this technology are going to be probably a very useful tool in those settings.
All right, thank you very much. So we've reached the end of our time for questions for today. I'd like to thank Dr. Fabio Luciani of the University of New South Wales, Sydney, for taking the time to present to us his talk on the use of single-cell multiomics for gaining insight into immune cell activity. Any questions that were not answered will be addressed by email if able. This presentation was sponsored by BD Biosciences in partnership with Wiley's Current Protocols. This webinar will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section under resources. Thank you for attending.
Thank you.